Well, thank you, and welcome to this year's McCosh Lecture here at Queen's. This is the year that we celebrate our 180th anniversary, not just as a university, but as an anchor institution that makes a real difference in Northern Ireland. And I guess we're proud not just of our convening power, but also the impact we have on our community. And that convening power is reflected here in having a meeting such as this with a guest speaker such as the eminent Irene Tracey. Now, we pride ourselves, therefore, not just in that academic excellence, but in the impact we deliver. This year, the theme of the McCosh Lectures, understand, are about exploring religion in the human. And speakers from the UK and Europe have considered the various ways in which religion engages with changing and contested definitions of the human, and offering perspectives gained from theology, from cognitive science, from anthropology, and from artificial intelligence. So it's a real great pleasure then to have Professor Tracey join us today. As you know, she's currently Vice-Chancellor of the University of Oxford, but she's also Professor of Anaesthetic Neuroscience in the Nuffield Department of Clinical Neurosciences. And following her studies in Oxford, she spent some time in Harvard, then returned to Oxford and worked with Professor Sir George Rader. And she was instrumental in setting up the Oxford Centre for Functional Magnetic Resonance Imaging of the Brain, which has now become the Wellcome Centre for Integrative Neuroimaging, of which she was director from 2005 to 2015. She then became head of the Nuffield Department of Clinical Neurosciences, a department of over 550 scientists, clinicians and scientists drawn from urology, ophthalmology and anaesthetics. So for over 25 years, her pioneering work has made a real difference to our understanding, and in particular to our understanding of pain and its perception and its processing. Her work is both discovery science and translational, and it's made a real impact, as I've said, on our understanding of the subject area. She's also an incredibly passionate advocate, both for women in science and for the public understanding of science. And I think we could use her expertise in the public understanding of universities, given some of the pressures we've been under recently. And I think she was guest editor of a Radio 4 programme quite recently too, emphasising that public engagement of science interests. Over the years, she's received, as you would expect, very many accolades and prizes. In 2008, she received the Patrick Wall Medal from the Royal College of Anaesthetists. In 2009, she became a Fellow of the Royal College of Anaesthetists for her contribution to the discipline. In 2015, she was elected a Fellow of the Academy of Medical Sciences. In 2017, she won the Feldberg Foundation Prize. The following year, she received the British Neuroscience Association's Outstanding Contribution Neuroscience Award. And in 2020, she was elected a member of the Academia Europa. In 2022, in the New Year's Honours List, she received the Commander of the Order of the British Empire for her services to medical research. So we're incredibly fortunate to have a speaker of this eminence, of this stature, deliver the McCosh Lecture, and it's my great pleasure to invite Professor Tracey to the stage.
Super. Well, thank you so much, Ian, for that very overly generous introduction, and to Andrew and Helen for facilitating my visit to you again. It was 2018 when I last came through. I was in another part of the building, and it's really great to come back. I apologise I've not been able to come back since. And thank you for being so patient. I was very happy to give it just orally, but it'll be so much better with the pictures. Can I just check that you're all okay to still listen for about 50-ish minutes? Is that okay? If you do need to leave, then I will not be offended. What I thought I would do in honouring McCosh and honouring what I understand is the range of disciplines represented here today, and of course at this great university, and congratulations again on your 180th anniversary, was to give you just a sort of foray through the field of neuroscience. So there's going to be a little bit of history about how we even knew the brain was where thought and perception and cognition arises, through to obviously the main topic, which is my work on pain, but not really covering most of what I've done on pain, which is a very medical, biological side, because I thought you might be interested in what I call my Friday afternoon experiments in pain, where I explore and went into the forays of looking at the intersection of religion and belief in pain. And so we'll spend a little bit of time there. And then at the end, we'll think about maybe consciousness and AI. So I hope that's okay. I'm hoping it's got a little bit for everybody. And there will be some sort of medically stuff there for those who are maybe medical, but it's not really a medical talk on pain today. So where does it all begin? Well, the human brain, it's a wonderful thing. I've spent my whole career working on it. Sadly, even though I love being a Vice-Chancellor, weirdly, the joke is often made that I was born for it because I work in pain. So I'm very good at giving it. I'm very good at receiving it. But I also do bring relief, I hope, occasionally in my role as Vice-Chancellor. But I can't be a neuroscientist anymore. And I certainly know that as an experimentalist, I won't be able, sadly, to go back to being an academic. So it was a big decision to pivot and do this role. And thank heavens I do really actually enjoy it. But I've had a wonderful career in this space, and that's what I hope to share with you today. And I still have a passion for just how the human brain works, because the thing about the human brain, which is special and different, and I'm bound to say that as a neuroscientist, and I certainly say that when I'm talking to my colleagues who work in cardiology or on liver, is, those are interesting organs and lots of things go wrong with those organs and we must understand. But the brain is what makes you who you are. And it's what makes societies what they are. And it's fundamental to our understanding of how we make our decisions societally, politically, etc. And so there's something just more about the brain that is intriguing and is enormously relevant. And indeed, beyond the sort of basic discovery, clinical of understanding the brain and what makes you you and how does the brain interact as social animals in societies, is an area that we really encourage more people to come into, because we need people actually outside of our discipline, people like you, to come in and think about, again, how we can map onto that. And that's a very exciting area going forward with lots of very impactful potential translations. But this is the brain, quick tutorial on how we got to this place. It's got these little things called neurons. They're the computational elements. It has white matter that allows the information to flow to different parts of the brain, where we've generally segmented function to different bits geographically across the brain, so like sort of highways shuttling the information around. The communication bit between those neurons, computation, is called the synapse, and it's a whole sort of soup of chemicals, peptides, neurotransmitters that helps keep the brain alive. It helps do the transmission across the synapse, and it helps modulate how that synapse is going to work in terms of sort of amplifying it, attenuating it, changing the flavour or the colour, if you like, of the synapse. So lots of ways of changing fundamentally how the communication is going, well beyond just electrical firing. So that's sort of in a nutshell what the brain is. It's just 1.4 kilograms. It's amazingly efficient. We talk a lot about AI, and we all know how computationally expensive it is. And it can't even get close to the human brain, which can pretty much operate on a Mars bar and doesn't need, huge amounts of compute time. And that's a phenomenal thing, just the actual evolutionary brilliance of what the brain can still do computationally. So we've got about 100 billion neurons. We have these other things called glial cells, which are cells that are called glial. It's Latin for glue. They were just thought to be the buffer, really, to just sort of glue all the neurons together. Largely ignored them for millennia. But in the past couple of decades, people have realised actually the glial cells are really important. So they provide a very important immune response, a very important controlling, modulatory, nutritional response. And again, a lot of different disorders of the human brain, certainly the ageing human brain, we're realising how important the glial cells are for that. So we've got these neurons, we've got these glial cells, there's several hundred billions of them. You go through these expansive phases where you just either make loads of them or you then attenuate them. And that's obviously during early birth, early development, all the way through to adulthood. And the best way to sort of put that into perspective is it's about the same as how many stars are in the Milky Way. And that's all inside your cranium. It's quite astounding, isn't it? Now, the brain doesn't sort of just get straight there, it develops. So these are three-dimensional etched pictures of how your brain went from birth through to about 20 years old. And there are two main things to observe here. The first is it gets bigger. The second is it becomes very folded. And that means you've got 20 years pretty much for your brain to be sculpted and shaped as a product of the environment in which you've grown up. And we're only scratching the surface now about how important that is. Nutrition, physical activity, educative background, stress, all these factors that are part of your environment and your nurturing, combined with obviously the nature, the genetic side, and will very powerfully influence what brain you end up with as a 20-year-old. And therefore, whether your brain has resilience in it or what we call vulnerabilities, and how that potential will play out then for the rest of your life, where there's still opportunity for sculpting and shaping. And this is quite politically explosive. There are some huge data sets being collected now, kind of like UK Biobank, very much at the adult brain, but again, collecting data about lifestyle, socioeconomic status, opportunities, genetics, etc., and being able to then make some causal inferences about, or correlative influences about how the environment might shape the nature of your brain and how that might lead to predisposition or vulnerabilities for then having addiction propensity or developing conditions of the brain in terms of, say, dementia as a major big one. It also explains, if any of you have teenagers, what goes on during adolescence, because we all know that they can be a nuisance. I've got 3 children, thank heavens they're all out of adolescence now in their 20s. 2 of them are historians, go figure. My husband's a physicist and I'm a neuroscientist, and we produced 2 historians who I'm very proud of. I got 1 physicist out of the 3. But they during their adolescent period, they can be a handful. We all know that. And there's quite a simple explanation that, is still a working theory. I don't have a laser pointer, so maybe you can just see my little mouse. Can you see my mouse there? If not, well, I'll you can see that graph. And what you're looking at there is, with age, you've got a change of growth and development of what we call the limbic regions, old brain, real primal old brain, fear, lust, reproduction, go find your mate, that type of stuff in your brain. And then you've got the prefrontal, which is all your high order executive, keeping a lid on things, controlling, making good decisions, rational decisions. And what you can see is there's a complete disconnect with age that hits at adolescence, and that you have an explosion of your old primal brain. You're motivated to go find a mate, and you will take risks to do it. And you haven't got a developed enough prefrontal to just keep it calm. And that's why adolescents, they can't help themselves. This is their brain. And in fact, colleagues of mine, Jones and Casey, this is quite old data now, but they would represent adolescents who've been joyriding or having criminal convictions in courts in the US, and try and get them off on the basis that, it wasn't like a free will thing. They just can't help it. They've got these brains that they just can't control, and therefore they shouldn't be, prosecuted for that. Now, lots of discussions we can go into, but, I just shared with you, to remind you how important this development phase is. And that's something, again, that's really very special about the human brain. And it provides a very interesting opportunity for us as a collective, as a society, as peoples, to think about how we want to shape people's brains going forward and what consequences that might produce. Well, we didn't know the brain was the seat of reason and thought. In fact, if you go back to some of these very old figures, right back, from the beginning of, sort of the early, 100s, we have these gorgeous pictures that, if you can see, very crudely just sketch inside the brain 3 areas which are like gaps. And it was thought that these ventricles, these parts of the brain which are understood to have basically sacs of fluid in, were clearly the important things for being able to bring information in from the eyes, bring it in then to where you're going to think about what does that mean, what's your cognitive decision about what does that thing mean that you've just seen, and then put it into memory. And it sort of made good sense that maybe is how it all works. And there was lots of fabulous further work done by Leonardo da Vinci, who would make wax cast of ventricles, then erode all the neurons and the glial cells away and have these beautiful 3-dimensional wax casts of the ventricles, because they were the important thing. This thing where the fluid was that was flowing around and probably could connect with the rest of the body made good sense. And the beautiful picture in there was a lovely exhibition of Leonardo's drawings in Buckingham Palace several years ago. So I went down to take some pictures and see it, because it's just so inspiring. It's such a huge part of sort of the neuroscience story. But it still wasn't quite sort of clear whether that was actually where reason was and thought was. It was a nice idea, and you can see it back in these very ancient pictures. And even if you look at a quote from Shakespeare in Merchant of Venice, you can see still the debate was raging, even in the mid-16th century. Tell me where is fancy bred, or in the heart or in the head? Because actually the heart then ended up becoming the dominant view as to where probably reason, perception, thinking was. Makes good sense. Got this organ right in the middle of you. It's got all these tubes that can connect to the rest of the body. It's got this fluid that flows around. You know, if I was back in the 16th century, I'd probably put my money on the heart being the one that is able to talk and feel things, the skin, and then make the muscles move and have you as a behaving living organism. So that went on, and we were still sort of not quite sure what was going on. And then, of course, along comes René Descartes, and he did a wonderful thing at that point in time, which was great at the time, still a problem, I'd say, in modern medicine now, is that he obviously came up with the concept of dualism. It's very important, actually, in my whole career, Descartes, because he did this, which allowed neuroscientists to go into the brain, start chopping around, having a look. But he also was quite influential in the pain field, which we'll come to momentarily. So what he did is he split it, and it was a very convenient little trick that got the church calm, and it meant that we weren't sort of stomping around in the sort of footprints of God and going into areas that might be considered the thinking around the soul and moral decision making, because there was the physical brain, which then was thought to be okay to go and sort of just chop out and do histology and work out what was going on. And then there was the mind, and that was something beautiful and sacrosanct. And we were not going to touch the mind, we're just going to go with the physical bit. Great trick, opened up the ability to do anatomy and all these other things, which moved us on our way in understanding. But we still have a very dualistic thinking. I'm not a philosopher, and although I have many conversations with philosophers in the context of pain. But it is a problem that we have a culture and a society where if it's in the mind, it's not real. And subsequently, patients and others are then treated slightly second class because it's not real in the physical sense. And we have a bias in our society towards physicality of something, whether that's structural, neurochemical, something you can measure, something you can see, something you can touch. The mind is something different. Well, as a neuroscientist, that's just bonkers. The mind is physical. It, you know, is executed through neurochemistry and neurophysiology. It's one and the same. But it still is quite rampant in society, and it's certainly rampant in the medical profession about this sort of dualistic thinking. And it does lead to, I think, oftentimes poor treatment for patients in certain conditions. So let's go back to sort of chopping things up. Great former professor of natural philosophy in Oxford, Thomas Willis, he would loiter down in Abingdon, which is a town just south of Oxford. He would loiter where criminals who'd been stealing things were being hung and executed, and he would collect the bodies and he would trundle them back to Oxford along what's called Dead Man's Walk behind the college that I was at and used to run, and then working with a young assistant, a medical artist, Christopher Wren, the very same, who did our beautiful Sheldonian Theatre and St Paul's Cathedral. He would do these beautiful drawings and sketches of the brain, both the vasculature, the blood vessels that feed the brain, different slices, labelling things by size and colour and shape. And these beautiful books, the Cerebri Anatomy, they're still in St John's College in Oxford. This is the little alleyway that they would do all their work in just here. And this is now one of the most popular Thai restaurants in Oxford, the Chiang Mai. It's absolutely lovely to go and have pad Thai in, but it does have this sort of slightly macabre history, which is where all the bodies were, and they chopped them all up and did all these beautiful works. This is a very modern interpretation of what's a very famous thing that Willis did, which is the Circle of Willis, which you can see here, and a very clever plumbing system in the brain, which allows a bit of redundancy. So if you have a stroke in one or a blockage, it can shoot around and make sure it can feed other parts. And we can image that now with much finer resolution back then. So we're starting to get a better sense of the anatomy. We then went through a slightly crazy era, I call it the era of folly, where a Viennese physician noted in the mid-1700s and 1800s that people that he would be having dinner parties with who had particular characteristics, they might be particularly like expressive, or they might be particularly thoughtful, or they might have great memories or whatever, and it seemed to relate to something about their physicality in terms of their skull, protruding eyes, bigger forehead, slightly odd shape there. I mean, honestly, but you know, I still teach the medical students in Oxford. I do several lectures a year, and I remind them, much as we would laugh at that concept, and we'd laugh that for several hundred years, phrenology took hold. The Phrenology Society of London only disbanded in the 1920s. How can a theory that's evidently just not correct persist for virtually 200 years? And yet it can. if it's credible enough, and you go and devise all sorts of wonderful contraptions and devices to measure it with ever finer detail, and you find the correlations you want, you can keep propping up your theory for a long time. And it's something that's a really good lesson for our young students to learn, that, you know, whilst we're in a world of disinformation, facts and alternative facts and what truth is, truth does evolve. And it doesn't mean to say it's not that you were lying or not being an expert. It's just that there's always a journey and a process, particularly in science. Things are very rarely binary and just static. They're always improving and upgrading. So this made good sense, and people could find evidence to support it. Until then, we come to a classic thing that happened with Phineas Gage, which you've probably heard about in the early 1800s, which sort of disbanded that whole phrenology theory, because this young gentleman who was working on the railway tracks, where they would be blowing things up in order to remove, obviously, you know, granite and stone to lay the tracks down. Some of these iron rods, which they were rodding in an explosion, shot off and went straight through his eye and through his brain, just ripped out half of his brain. And overnight, he actually survived, but he became a completely different person. And so that was a very, you know, clear causal relationship between injury and personality outcome, what makes you you. And so the location that this stuff that is, you know, the grey matter, the white matter, the glial cells, it's not just padding to support the ventricles and look after it. This is the business end of things. And if you damage it catastrophically, then you will have fundamental changes, not just in the ability to move or see, but in things that are even more complex, like what makes you you. And he became a very different person. And then it was a sort of free-for-all in the days that you didn't have to get ethics approval. Canadian surgeon, very famous one, Wilder Penfield, whilst going into the brain to take out a little bit that was seizing a lot to cure epilepsy, would prod about and have a go and see what would happen if I just stimulated that or recorded from there. Because the amazing thing about the human brain is, once you get beyond the skull and the skin coverings and everything else, and you're actually in the brain, there's no pain receptors in the brain itself. So you don't feel anything. And going in, it's agony so that's why they put the patients to sleep, open up the skull, and then that bit's all done without any pain. And then once they're open, you can prod around and they won't feel anything. So you can record live. You can say, if I stimulate this, what goes on? And the person, oh, I'm remembering an incident here. And then you can map, if I touch you here, which bit activates. So he did this very famous thing called the homunculus, where he mapped which bits of the brain map your body representation, which bits of the brain map your physical movement. And there's these beautiful strips that say your sensory cortex. You've probably seen the funny-shaped man, where the representation of how much brain is given over to detect something that you know you've got fine-grained detection on, like your fingertips, has a big bit of the brain. And other parts of your body that you know you have poor sensitivity, say the trunk, has a tiny bit. Lips big, tongue big, other things to do with reproductive organs big. And so you have this man that's a very funny shaped man that maps onto how much of the brain needs to be given over to give you the relevant resolution you need to detect things in the environment and to be able to, you know, feel things with the sensitivity you need. So really important experiments, less easy to do now because they're just rightly different ethical controls coming in, but revolutionary. And the most famous thing for Penfield is the Penfield homunculus for sensory and motor mapping the human body. But again, just consolidating that, you know, the grey matter has this function, and it's geographically got different functions in different places. So that then led to the idea that, well, wouldn't it be great if we could develop in vivo, what we call in the living, rather than having to do this? Wouldn't it be wonderful if we had tools where I could take you, just put you in something and actually measure which bits of those brain are becoming active when you hear, smell, taste, touch, feel pain, remember something, see colour versus black and white, movement versus static, more and more going into, you know, the vision perception. So if you think about what you are, you have several basic perceptions, and most of them are conducted through organs in your neck and up. So your eyes, you see, you hear, you smell, you taste, and then you have the touch system, which is obviously all your skin, and then obviously internal things that you can feel. And that's how you communicate with the world. And you absorb all that, the brain works it all out, you decide in the context in which you're experiencing it, what does that mean? How do I want to interact back out? And then you execute a decision, and then you move in order to interact. And your basic daily job is to largely seek things that are nice and avoid things that are not nice. And that's sort of the little cost balance decision making. If you really think about what you're doing all day, is what you're doing all day long. And the value of that cost balance can change. If you're thirsty, a glass of water is more rewarding, and you'll be more motivated to make the decision to walk over to the tap and get it. If you're not so thirsty, the value of that is not so much, so you're not so willing to make the energy cost to go and do that. You're just doing that all day long. Very back to Bentham, in terms of cost balance. So those are the sort of, basic sensors that you have in the human brain. And what we wanted to do is try and map them. And this is where sort of imaging arose. And it goes back to another great physiologist in the day. Again, we all claim Sherrington. London's claims and King's, Cambridge, Oxford. He's probably at Queen's as well. I mean, we all claim Sherrington. But he was amazing. He did lots of things in pain as well, like Descartes. But he also pointed out that the brain is highly perfused. This is a cement three-dimensional structure of your brains. If I perfused your brains, if you were dead, and then perfused your brains with cement through all your capillaries and blood vessels and then rotted away all the neurons, that's how perfused your brain will be. It's one of the most highly perfused organs. It's extraordinarily beautiful in that regard. And what he showed in some very elegant experiments with very basic kit, not like the fancy stuff I have, really brilliant experiments, that when you have a perception like seeing or hearing or smelling or tasting or touching or doing something like thinking or making a decision or having a memory, it isn't just lots of blood flowing up to the whole brain. It's preferentially given to that bit of the brain that's subserving that function. And he was able to prove that it was a local delivery, not a global delivery. And that 100 and odd years ago gave us the basis, subsequently many years later, to take, and I'm not going to go into how imaging works, use magnets and large MRI machines to exploit that phenomenon and be able to detect with a sort of bells and whistles version of a bog standard MRI where in the brain blood is preferentially flowing to feed the parts of the brain that are subserving the particular function that you want to understand. Where in the brain does this happen? And is it broken in a disease? And that was the birth of functional imaging. Now what we do with most imaging is we collect all sorts of different data that tells us about the structure, the function, the white matter connections, how healthy are they broken down, what's the natural conversation going on in the brain even at rest, what's the chemistry, and then what's the brain activity in response to pretty much anything that your imagination can come up with in terms of the questions you want to ask. Again, for time I'm not going into, but we've done, again, my Friday afternoon, utilitarian, non-utilitarian decision making. When people tell you one thing, we can disambiguate, did they really struggle if you threw somebody off the bridge to save the trolley? Or did they not think twice? Now, they'll tell you, it's a moral decision, I was struggling. We're doing the brain imaging. No, you didn't think twice, you went for it. And it goes with psychoticism. You can explore stuff with imaging that the person doesn't want to tell you because it's not socially acceptable to tell you. And that's the beauty. And which is why there's this whole, discussion and debate, and I've written, reviews on this about the use of imaging in courts of law in other areas where sometimes, you might want to reveal what actually is going on as opposed to what the person wants you to think is going on. So we've got these wonderful tools. We're very, very lucky to have them all. We've got other tools as well where we can look at more of the resolution, the receptors, all of which are very useful for the more medical applications of trying to understand the human brain and how it sadly does go wrong as you get older. But let's go back to Hippocrates. So I've just told you it took us until pretty much the 19th century to work out the brain was where perception arose and where cognition is and where memory is and where decision making is. But actually Hippocrates, who is just such an extraordinary individual, 400 BC, put the brain as the centerpiece, probably of where these very subjective things are, like your grief, your sorrow, and he put pain in there. And this was well before we knew that the brain was important. And that's really, really extraordinary. So we've got these tools. We now know the brain's it. We can start to really take on beyond just, understanding vision and all its richness and hearing and etc. I've spent my career trying to understand, a really complex experience that's both sensory and emotional, highly subjective, highly private, and that is pain. And with that, you can look at, again, indirectly, empathy, you can look at suffering and consciousness as well, we've worked on. And of course, it's just part of the human condition. It's part of the animal condition to experience, sadly, pain and suffering. We see it all the time. We see tremendous overlaps in the brain in an empathic state versus a physical, painful state. We get to the heart of issues around biases in society about the physical pain versus emotional pain or psychogenic pain, and we have judgments, which we'll go into a little bit about that. And it's been a great field, and we knew nothing about it until we had the ability to bring these tools in to start to tackle this problem and bring some objective mechanistic understanding of what underpins and constitutes a person's private subjective pain state. We are not objectifying a subjective state. That's a bonkers, ill-thought-through statement. You can't objectify something subjective by definition, but you can certainly objectify and have objective information that can help you understand better why that individual is having that particular subjective pain experience. And that's what we've largely done, but that isn't what mostly I'll talk about today. Just give you a few examples. But in terms of where we are on the pain field, acute pain is great. It is your warning system. The whole point of having a body system that is able to experience things in the environment that are going to hurt you is it warns you to get away from them, remember what caused them, so don't touch it again. And then if you are damaged, to attend to it so that you heal. That's acute pain, that's great. Chronic pain is just the system gone wrong. It is something that we define as something that's still there 3 to 4 months after normally you would expect the tissue to have healed and got better. And if it's still there, you're defined as having a chronic pain state. And we now just very recently have defined it as as a disease in its own right for 7 conditions being listed. A completely different way of thinking. Chronic pain not being just a symptom of ongoing acute pain, but in these 7 conditions, it's now become something other. It is a disease in its own right with its own mechanisms, its own problems, and those we have to target, not the acute pain mechanisms, because they're completely different. And that's where we failed. That's why we have most people finding that the medications don't work. If they do, it just takes the edge off. So for 3/4 of patients, none of the medications work. We've completely failed to develop novel and new therapies to target things. It is much better now because we've had a paradigm shift and we think about chronic pain in its own state, not as a symptom, trying to fix the acute pain and thinking the chronic pain will go away. That model has failed us. So now we're understanding chronic pain and the mechanisms, targeting those mechanisms. In the next decade, there's going to be wonderful new therapies coming out targeting completely new biology around chronic pain. It'll be much better for patients because it is the biggest medical health problem we have in the developed world, and it costs a fortune in society for the treatment management and people not going to work because of their migraine and their backache, etc. So it's a huge, huge problem. As I say, lots of discoveries being done. So every single perception you have, whether you see, whether you hear, whether you smell, whether you taste, whether you touch, it's all built on what we call a 3-neuron order system. The first neuron out there is just talking to the things in the environment to pick up the information in. That's transduction. The second one is transmission. It's getting that information, shuttling it in to the central nervous system, the spinal cord and the brain. That we call transmission. And then the final neuron is sort of up in the brain to the network to give you the perception. So whether you're looking at hearing or seeing or smelling or tasting or touching or feeling pain, it's all built on that 1, 2, 3 order neuron system. And we know a lot about the transduction. David Julius and Arden Pataputin got the Nobel Prize for this in medicine and physiology a couple of years ago for discovering all the things in the environment all the receptors in you that can communicate to the 3 main things in the environment that hurt you, which fall under 3 domains: temperature, chemical and mechanical. If you think about it, those are the 3 categories of things that hurt you. And they've worked out brilliantly the actual business end of what talks to things like a bee sting or chili pepper hurting you or cutting mechanically with a barbed wire or hot coffee on you. So that's bits worked out. The transmission bit, sadly, that's been worked out because of patients. You can see this middle picture here, smashed legs. Yep, those legs are completely smashed. That person didn't feel any pain. Jumped off a building, smashed their legs, didn't feel a thing. So for many, many millennia, we've known that patients can have this thing called congenital insensitivity to pain. They just don't feel pain. And they were basically freaks, and they were used in circus shows and performances. They would die because although they didn't feel pain, they didn't have the warning system to tell them they've completely smashed up all their internal organs. It's bleeding internally, or you've got an infection and it's going to get septic, so they would die. These days, wonderful physicians look after them much better. And now we've been genetically profiling them, and my colleagues, and discoveries have been made about why is it they don't feel pain. It turns out one of the major genetic explanations for that is they don't have the working kit to send the signal in. So they got the transduction kit, they got the perception kit, but they can't get the signal in. So the transmission bit's broken. This is a target. These two transduction transitions, these are new targets now we can go for. Because if you've got a patient who's constant ongoing pain, you can target now what we've learned from this and stop the transmission going in. Now you've got to titrate it because you don't want to make you a congenital and sensitivity to pain patient. So you want to take away the bad pain and keep the ability for you when you touch a hot pan to withdraw your hand. And that's turning out to be a bit more tricky than they thought in terms of drug discovery, but they're still going and hopefully that will work. And then the perception is the bit that I've worked on, and we will get to the belief religion bit in a minute very soon, I promise you. So I've worked on perception. It's hard. It's been really, really hard. We still don't know the bits of the brain. You know, I've got a very good working hypothesis about if I was going to build a robot to experience pain, what bits of the brain I'd put in. But it's still, that's my hypothesis based on many years of doing experiments. There's still not, consensus yet. We're still doing the research and the discovery about which parts are really important to give you the perception of pain. That's different to vision. That's different to audition. That's different to taste, where we're very clear which bits of the primary cortex. We're close with pain, but we're not there yet. Because of this subjectivity and this societal bias and this mind-body split, it's a real problem. So I challenge you. You've got these two x-rays here, these two hands. You're all going to be very comfortable that hand, if the person told you that that hand was extremely painful, you'd be very comfortable with that answer, yes? If you saw that hand, you'd be very comfortable that a woman, say my age, late 50s, says no pain. Yep, because that fits our wonderful lifelong medical model. I like to see something structurally before I'm going to believe it, right? We're just biased that way. So pain here, no pain there. You're probably okay if I said to you, your elderly grandmother's got this hand, but she says it doesn't hurt at all. You'll be, oh yeah, that makes sense. She's really hardy actually, and they're built like tanks, and she's always been stoical, etc. You're perfectly comfortable with that because that's sort of societally acceptable. Again, let's go to another woman of my age. She tells you that hand is agony. You are not. I promise. I bet you, deep down, you're not going to be as comfortable with that. You think, really? What's going on here? And that's the bias that plays out every day in a GP surgery or in a hospital. Physicians are absolutely fabulous and brilliant. It's not their fault, but we've all been influenced by our nurturing and our model and our need for the physicality. And therefore you start to think, well, now, is it really that? Maybe she's depressed, maybe she's anxious, maybe she's neurotic, maybe there's something going on. Everything but, maybe I don't actually understand pain very well and it's more complex than this, and I can't possibly make a judgment call about her private pain experience based on what I'm seeing on a crude x-ray. That's the right answer. And so you can't possibly. It's just too complex. Even just what I've shown you about the transduction, the transmission, and the perception, it's a highly complex, highly evolved system. There's so many points where things can be changed up and down, and then even which bits of the brain get activated can be altered. You can't possibly know what's going on here from what you measure out here. That is an impossibility. And again, that's something I teach the medical students at Oxford, and they get drummed into that. And of course, you bring your biases. Blokes are brought up that you have to be macho, there's no gain without pain. We have, people like Dostoevsky and others in our great literature telling you can't really be a great intellect unless you suffer. Midwives tell mothers it's great for you to suffer in childbirth. It's good for mum and good for baby. Well, I've had 3 and I was not happy with that, so I had epidurals every time. What a load of nonsense. I'm a practicing Roman Catholic, but my goodness, we've got a lot to answer for there in terms of religious and martyrdom and suffering. Actually, the church has been genius, and I'll come to that in a minute in terms of how it's handled it. But we've got all these things that we're bombarded with, which set our attitudes to what we think somebody's pain should be, based on your own journey, but also all these bombardments. And that makes it tough for the physicians, makes it really, really hard for them to sort of, do their job with objectivity. So how do we measure pain? Well, we ask people, how painful is it? So you can get a sense of, if somebody's been in chronic pain for 20 years of their life and they've lost their job, it's completely become their identity, their marriage has broken down, etc., etc. And then we ask them to say, tell me about all that suffering on a 0 to 10 scale. Are you a 6 or are you an 8? How can you possibly capture that level of suffering and impact on these scales? But we do the best, and really good intuitive physicians do their best to combine different ways of measuring pain and getting more qualitative and quantitative data, looking and observing. If they're asleep or comatose or demented or a baby and they've got no language, then you have to use indirect ways of looking at it. But none of these are very satisfactory. And they've all, we know, led to why we don't have good new pain drugs out, failed legal disputes, insurance claims, etc., etc. And it's because it's a hard problem. Descartes, again, told us, back in the 16th century, you put your foot in the fire, there's a nice one-to-one mapping to basically activate the brain, and it's a simple direct line. Well, I'm sorry, Descartes, but that's just not, sadly, it's not like that. It's a highly what we call nonlinear. It is not a simple one-to-one mapping. And the journey from transduction to perception has all these points where that signal can be turned up, down, routed, and then access completely different brain networks to change the quality, the intensity of what you are experiencing. So it's this incredibly emergent, multidimensional experience. It's completely malleable. It's subject to lots of influences that are all brain-based, like your mood, the context, and the cognitive state, and that produces that non-linearity. So hopefully now it's really clear you can't possibly judge what pain somebody should have based on a very crude X-ray of the periphery. It's just more complex than that. So I've spent my whole career basically working on the gap between transduction and perception and explaining the nonlinearity by looking at perception and trying to understand how does that nonlinearity arise? How is it that you can have no pain, no damage, but huge pain, huge damage and no pain, and everything in between? And where are the points of volume amplifiers and changing networks? So that you change the attentional state of somebody, distract a child having a next vaccine jab. I do marathon running in at the end, absolute in agony, very strong goal. How do I block it? How do you get through that? Grief, empathy, depression, anxiety, how do these all influence? So if I took any of you to my scanner and I gave you a pinprick or a burning pain, you would activate this incredibly complex set of brain regions. Don't worry about what they are, it's not relevant here. But if I changed your attentional state, and I distracted you, or I made you attend, or I changed the context, or I made you sad or anxious, I can completely change your pain experience. Both how subjectively you feel about the same burn, and it will be reflected in a different pattern of activity. And I can make it go up and down. I can even make pain pleasant and hedonically flip it. It is really easy to manipulate it, because if you just know which bits of the brain to access the network, you can just completely change how you would experience the same input. So you get the same pattern. You can see a little bit of changing activity there to the same burn. So if I change the thing, you'll change the way those signals are processed. They'll come in, they'll activate different brain regions, and that will change the experience. It's as simple as that. Highly nonlinear. So just to really drive that one home, these are two extreme examples. On your left is a person hanging themselves from body piercings. There are people who do this for a hobby and a fetish. They experience no pain from that. In fact, they quite like it. And that's some other work we've done, but we won't talk about that today, the pain-pleasure one. So, huge amounts of tissue damage, no pain. On the right is a person who stood on a 12-inch nail. It's a builder in London. Some of you might have seen this picture before. Screaming in agony, went into University College Hospital London, and when they took away the laces and they opened the boot, they saw that the nail had gone between the big toe and the second toe, hadn't cut anything. And straight away, pain gone. It's not fake, it's not making it up, it's not unreal. I'm not sort of, classist about whether pain is got a physical injury or whether it's generated by brain networks. At the end of the day, pain does not exist until the brain produces it. It's no susception until this point, because pain is a conscious emergent experience. So I don't care whether something comes in to drive those networks or you can switch those networks on. That's pain. It emerges in here. So again, societal biases are, we like the physical, we like to see blood, guts and gore, but you've got to get over that, because if that person is able to generate the network activity, they are having a pain experience. It is completely different generator, and your job is to work out which one it is. You shouldn't judge it as A class, B class pain. So he had pain, the fear, the fret, the anxiety. Of course, once he opened, then, not there, that's good. So no damage, huge pain, huge damage, no pain, everything in between. And that's what I've spent 30 odd years, the team have spent 30 years doing lots of experiments to work out all the mechanisms that then have lots of, hopefully, translational relevance. For the medics in the audience, this is the nut job of sort of what we know now, just very quickly. The big problem of what holds people in chronic pain Instead of when the nerves in your periphery that detect things that are sharp pain, like pinprick or mechanical or thermal or burning, normally when you damage a nerve, it doesn't work. You damage your optic nerve, you don't see. An auditory nerve, you don't hear. When you damage the pain fibres, as we call them, weirdly, they switch on and they just start what we call ectopically firing, just sending signals in. It's a constant firing of nerves. Then the amplification of those signals occur because the central nervous system reacts to that and then sets off all these amplification. And then if you don't deal with it, because we haven't got good drugs to treat with it, that then takes, on average, most chronic pain patients will be at least 7 years of their life with chronic pain. That is a big chunk of your adult life lost. That's why it's such a huge cost and suffering for people. But the constant barrage and amplification sets off rewiring and maladaptive plasticity, which we're very worried, 10, 15, 20 years in pain, do you just get locked in? And it's really hard to get the people out. And then, of course, the first question goes back to where we began. Who was the 1 in 5? Because 1 in 5 of the adult population have chronic pain with that definition I gave you. That's a staggering number, but 4 out of 5 do not. So what makes that 1 out of 5 tip if they've got diabetes and they've got nerve damage with the diabetes? Why does 1 in 5 get pain with it and 4 out of 5 don't? Does it go back to the vulnerability question? So some of the very latest work that we were doing before I switched to this job, and we're just finishing off a few papers, the last few doctoral students to come through, is looking at the vulnerability. And is there something predisposing about your brain that you were always going to go chronic? And we could tell you ahead. So we did a range of experiments, pre-surgical for new hips, where we could predict who was going to have a good outcome and who was not. People who had cancer and were going on to chemotherapy, which can damage nerves and produce nerve injury pain, a very classic what we call neuropathic nerve injury pain. We could predict who was likely to develop nerve injury pain based on theories of what we know goes wrong in the brain networks about vulnerability. So that's just a sort of quick whistle-stop tour. And I want to finish by just talking about how does belief. I'll go to sort of what I call my Friday afternoon experiments, because that's sort of probably more of interest to most of you here. So here's just a couple of experiments we've done, and one experiment that led to then this religious study we did. So how does belief intersect at a mechanistic level? Well, the first sort of way to think about belief is a big concept. Obviously, there's a religious element to it, and we'll come to that with an experiment we did. But one of the ones that led to that, in terms of giving us the anatomical hypothesis, was the belief that you can control your pain. And so in this situation, subjects were administered pain, and they were told that there were several scenarios. One, somebody in a room next door is going to decide when that burning stimulus is going to be turned off. In another context, you could decide when to hit a button and it would turn off. And in the third condition, it was a computer would just randomly decide. And that's just happening. They're getting told it's you, it can stop, it's the machine, it's the person in the room next door. What they didn't realise is that very cleverly they were being yoked to everything. So they actually got the same duration stimulus in all those conditions, but they just didn't believe that they had the control of that. And so we're looking at how belief intersects with controlling your level of pain. And you can probably intuitively guess what's going to happen in terms of how they felt about the pain, because a lot of what we do in imaging is basically just prove objectively what you sort of intuitively know is probably going to be the case. And so what we showed was, indeed, the pain intensity rating. If you're controlling your pain in the self-controlled one, you rate it as significantly lower compared to when either the person or the machine is doing it. So you get extra analgesia just because you've got the control over it, because you believe you've got control even though you haven't. Your anxiety ratings are much better. It brings down this anxiety. And then I just want you to remember one bit of the brain, this bit called the anterolateral prefrontal cortex or ventrolateral, right, that very high order out in the frontal cortex, the bit that takes a bit slower to develop as you go through adolescence. This is the bit that seemed to be really important in that condition where you had that benefit of analgesia or pain reduction because you were controlling the pain. And then people were stratified to work out whether they were a bit of a control freak or not. We all are on the spectrum somewhere, and the wonderful thing of working with psychologists is they have questionnaires for everything. So they've got these questionnaires, and marvellous questionnaires, which is your basically self-control. It's basically, are you a control freak? And you can stratify. And what you can see is, if you have a high sense of control, you really are a controlling individual, and you are put into the self-control one, well then of course you like that a lot, and that's going to give you that nice activity of this region of the brain that produces the analgesia, this frontal bit of the brain. If you are a person that, again, is out here, high self-control, are you seeing my little mouse? No, all right. So if you're a control freak, you're at this end of the axis, and you can see you don't activate that bit of the brain in the situation where the machine or the person's controlling it. You really don't like it, and you don't get the benefit of the analgesia. So it goes with just things about you that, again, sort of intuitively makes sense. But maybe counterintuitively, if you were a physician working with patients with cancer on a patient-controlled analgesic pump, you'd think, a bit of a control freak, that one, not going to give them the pump. That's exactly who you should give the pump to, because they really like control, they've got control, you should let them have it, and that will give you activity here. Now, why am I telling you all that? Well, this little bit of the brain is really interesting, and we know that this is this region of the brain where you can sort of dissociate yourself, if you like, from the emotional content of what it is you're experiencing, which is something that pretty much hurts. So we sort of have other data and studies that would suggest this is quite an interesting little bit of the brain. And of course, this seemed to be really blasting in that situation where you've got that strong sense of belief about being able to control your pain. And so then we know that people who are religious generally have what we call a high external locus of control. This questionnaire that stratified whether you're a control freak or not stratifies based on this whole sense of locus of control. Are you a high external locus of control person or internal? If you're an internal one, you're a control freak. If you're external, generally, I'm making sweeping generalisations and I'm nervous here in front of this audience of scholars from theology, but generally, people who have a religious nature have grown up and nurtured and shaped their brain to be very comfortable with an external locus of control, a benign God. Fate, a God, somebody who looks after me. And you've therefore wired your brain such that in situations, you harness that to benefit. So our hypothesis going into this experiment, we're going to come back to the Pope in a bit, the new one, was to do this particular experiment where we wanted to look at the intersection of religious belief on pain and analgesia and suffering, with the hypothesis that people who are religious will be able to activate that little bit of that frontal cortex because they're highly external locus control. They're able to dissociate themselves from the emotional suffering component of it because they've used things like imagery their whole lives to do that, and that they would get the benefit of analgesia. So we did this incredible experiment where we had functional imaging, galvanic skin response, which is like an independent lie detector, if you like. We had a group of very, very well phenotyped, not just ticking a box Catholics, but practicing Catholics, very prayerful, going to mass regularly, etc., and atheists. We had these very matched visual pictures that we worked with artists and art historians to tell us about how we could match these two beautiful pictures best, so that they were all sort of visually loaded and content, etc. And then we did a classic what we call 2 by 2. So the religious group and the non-religious group all got to see the Virgin Mary, and they all got to see the Lady with the Ermine. So everybody got to see everything. And they all got the galvanic skin response recordings, and they all got their brains imaged. They also all got electrocuted whilst doing that. So we do what we call a mood induction, where you watch that picture for a bit. Obviously, that's quite meaningful for a Catholic. It's not very meaningful for the atheists, and vice versa. And then we are electrocuting you throughout it. And then we measure, well, how did you feel? What was your pain rating? How did it affect you? What was your anxiety, etc., etc.? And our hypothesis was the religious group would have the benefit of the picture producing analgesia, and they would activate that bit of the frontal cortex and would get a galvanic skin response collapse, but the atheists would not. This experiment was interesting in so many ways. It was the only experiment in my 30 years that I've ever been told by the ethics committee, I can't do it. I have put balloons up people's rectum and down their esophagus and blown them up so it hurts. That's fine. You can do that. I've given people, doses of anaesthetic agents, put them asleep inside the scanner and burnt them. That's fine. Highly, highly difficult experiments. This one, banned. And it was because a person on the ethics committee, which I won't say what background they came from, but interestingly, they were very concerned that should we show that there was no benefit of the religious group, what was our post-experimental plan? To which, when I was rebutting this and going back in to say, no, you need to overrule this decision, one, isn't that unethical that you only seem to care about the religious group? What about the atheist group if we do show that there is a religious benefit? So you don't seem to care about them. And also, as a practicing Catholic, I wouldn't like to think that your faith is so shatterable that one experiment with me is going to sort of undermine you. So we got it approved and we could do the experiment. But then we couldn't get it published. Nobody wanted to touch it. This is actually, we've published in all the great journals. This is probably one of our most sophisticated, I think, and interesting experiments, but people were terrified to publish it. So when eventually it did come out, The editor said, I'm going for it. Put the Virgin Mary on the front cover, went for it. It was covered in the Radio 4. Sandy Totsvig did a hilarious thing saying, those Oxford academics are back torturing the Catholics again. Can they never give it up? And it was very funny, and it had a good spread, and it's lasted to this day. It was a very early experiment in this area of social neuroscience, asking questions that were of more of a social nature. And as I kept saying to the reviewers, it's not sort of about religion. We use religion because it was actually the easiest thing to control and do the controlled experiment for. It's about belief and something that's been a really positive influence in your whole life. I could have shown a footballer, David Beckham, I probably would have got the same answer. But the fact is, when we thought about what would be something that would really have influenced you in your whole life and shaped your brain to react in a different way in a context of suffering, actually, we went through education, went through, religion, parenting. Religion ended up being the easiest one, and it locked that hypothesis, and it worked brilliantly, because the upshot was it actually worked out. So the people who showed the pain reduction, surprise, surprise, was the religious group when they were looking at the Virgin Mary. Both groups preferred the picture that you would expect. Surprise, surprise. They activated the anterolateral prefrontal cortex, the religious group, when they're looking at the Virgin Mary. Their galvanic skin response came down, and they got analgesia. And it worked like a treat. So it's just a little example of, again, a different experiment looking at control. You get the hypothesis, you understand a bit more about the brain, you look at other studies and papers, and then you can start to ask, at that point, this is going back many, many years, and you look back now 20 odd years, you can ask a quite challenging question at the time. I didn't expect it to be that challenging. So for my poor postdoc, we then just left this area, because actually it was career suicide for her. Once we got the paper out, then we had to go on to other stuff, and she's been brilliant, since. But anyway, and now people, of course, are doing lots more of this type of thing. But there's a lot still to go in there. There's a lot to go in about how, again, in our context of all the beautiful neuroscience and imaging about decision making, how do you make decisions individually in a society? How do those decisions influence a moral decision-making versus not, in all the different contexts? You can do really sophisticated experiments. Trouble is, people like me don't really know the questions to ask. I suspect in this audience, you're the people that should be talking more to the imaging neuroscientists, building on what we're understanding about emotional parts of the brain that are core to belief, which is a very core animalistic thing. I'm going to just touch on that at the end about hope. These are very fundamental ways that we are wired. And, we've got the goods, we've got good understanding of the neuroanatomy, we've got some good theories. If we could couple it with people who actually have really deep understanding of the types of questions that would be interesting to answer, I think there is value in society to give the evidence base of just how the human brain reacts to that. So I think actually that can inform us in lots of interesting ways going forward that we probably do need to understand as to why people are so easily manipulated in terms of charismatic figures, and how we're so drawn to certain things or not, and why is it that we go tribal when the world becomes more complex? What is it about how we've evolved still very slowly compared to how the world's evolved in such a complex way? These are really profound questions that we need to understand to explain why decisions in terms of politics or whatever that are going on at the moment are occurring. And I think we've got a chance to do a bit more of that, and I encourage you to talk to your people to do that. Another big aspect of belief, which again is something we spent a lot of time on, is belief as it comes as a placebo effect. And this is something, again, that we've spent a lot of time working on. Just a little bit, just before I explain that, you'll need to understand is in the oldest part of the brain, which is that brain stem, so there's your spinal cord coming up, brain stem, and then into the brain. This is very old brain, shared across, different animals. It's got this machinery in there called the descending pain modulatory system. It's the bit that we've spent most of our work working on and understanding. It's beautiful. It's this extraordinary milieu of anatomy and neurochemistry that is exquisitely positioned in the brain, old brain, to talk to high, newly evolved brain, down to old spinal cord. And basically it's the gatekeeper to pain perception. It dictates how much of those nociceptive injury signals are going to get in. It controls at the entry point to spinal cord whether they're going to be turned up or turned down. And it exquisitely controls that and is influenced by top-down mechanisms. And you don't have that level of control for any other sensory perception, sight, hearing, taste, smell, but you've got it for pain, because actually it's really important that you have the pain that's appropriate for the situation you're in. So if you think about what pain's purpose is in the good sense, acute sense, you have an injury, you decide what to do. If it's a little pain, you won't even notice it. So the level at which the pain is in the context in which you're experiencing it will trigger your decision making as to what you're going to do or not do about it. And that's really important, that you make the right decision. And sometimes you're not going to make the right decision if the pain's overwhelming you, because the situation could be so threatening, you could end up being killed. So over millennia, we've built and evolved the brain system to have this extraordinary system of controlling and being a gatekeeper, so that in a situation where half your leg's bitten off by a lion, you just block it, you scrabble away. And the body's built a system to just kill it. You just don't feel the pain, because you're going to get killed if you don't, and you can't be distracted by the pain. So more regularly, you'll see that in sports, battlefield, fight or flight. It's that. That's why you have it. Again, like most things in the body, beautiful system like that, powerfully useful in these situations so that you make the decision that's right for the context you're in, and it allows you to do that. It gets hijacked in the disease state of chronic pain. So these two cops, good cop, bad cop. The good cop is what you drive when you're top down going to block the pain, run away from the lion, get that try on the rugby field even though in the scrum you've had your hair bitten off, all those sorts of scenarios that you play out. Sadly, the bad cop turns the signals up and makes it worse. And we know that this is really important for persistence of chronic pain. And we've done loads of work on that. And that's a big target for discovery and blocking. So this balance between good cop and bad cop. This is also important for the vulnerability. We now know I could take you all now, put you in my scanner, and you would all be at a different set point of having a bit more good cop or a bit more bad cop. And depending on whether you don't have a very good cop, you're more liable to get a persistent chronic pain state should you have an injury, should you get diabetes with nerve injury with that, go on to cancer therapy, etc. That's the working hypothesis, and that was the latest work that we were doing. So this vulnerability question, these things get set as you're from birth right through early toddler years. You're setting the point of how healthy your good and bad cop are. And it turns out we now know this is very profoundly important for deciding whether you're going to be the 1 in 4 or the 4 or 5, the 1 in 5 or the 4 out of 5, because of that. And it's key importance in controlling what's allowed in. So we've done a lot of work on that. And one of the great questions that we first asked, or not great, that's the field asked, not just our group, was, is this what you're using when you're distracted from pain? You know, distracting your child having the vaccine? Yes, it is. Is it what gets you over the line, you know, after you've got the goal and you want to do that? Yes, it is. So the next obvious question is, I wonder if it's the placebo effect. Now, the placebo effect, you can see these monks holding these yellow liquids over there, and that sort of symbolising the Beecher experiment. The placebo effect is something that has had a bad press for all the wrong reasons. The monks would come and sing these Latin chants. They're called placebos. That's why that's where the name comes from. But the reason the placebo word has a connotation of fakery and deception is the monks who'd come and sing these placebo chants were paid to come to your funeral and pretend that they were sad that you were dead. And that's why, for many, many years, we have this societal view that placebos are bad. Some of you will probably remember the placebo test to catch the patient out, to see whether they were really in pain, completely based on utter, almost as bad as phrenology, misunderstanding of actually what the placebo effect is. It is one of the most powerful analgesic mechanisms we've got in the human body. Frankly, it works better than any drug we've got currently to treat pain. It is brilliant and it is awesome. But the moment we actually really proved it was a thing, which was Beecher's great experiments in the war, we threw it out of modern medicine. We said, well, we've got to stop doing that. I don't know what was going on then. Everybody must have had a collective seizure. But right at the time when we proved it was actually real and good, we decided to completely throw it out of medicine, whereas actually really brilliant physicians, brilliant bedside physicians, absolutely knew trust, confidence in you and the patient partnership, managing expectations of the patient. This had an influence that was real. It wasn't in the mind, it was real. They just didn't have the tools to prove it. The church was fabulous at this. My own church was brilliant. You know, there's nothing working. So, you know, the more you suffer in this world, the more reward in the next. What a genius trick. You're just offsetting reward. And people were much better able to cope with pain and suffering because they knew the more I suffer now, the more reward I'm going to get. They set the reward systems on. It helps produce, they just didn't have the chemicals. Lotions and potions. It's all placebo. But they worked because, it's trickery how you elicit it, but it elicits a very real physiological thing. And the question with the imaging is Is it just using this system? Now we know what the system is. Well, the answer is yes, that's all it does. The very sciencey, these slides for the scientists or clinicians in the audience, but you can do spinal cord imaging, you can do brain imaging. It absolutely uses the good cop, and it just stops the signals coming in, boom, and it works as good as if you're squirting in a load of opioid. It's fantastic. But we don't use it in modern medicine because, the placebo effect. So very interesting. Anyway, so what we did was this experiment, which again, will be the last few I'll talk about, and then I'll stop and ask questions, was to say, okay, we can't use it in modern medicine because it requires deception and fakery, and the physician can't pretend to the patient, give them a sugar pill or a saline injection. But actually what a physician is doing is having a conversation with the patient, setting up beliefs and expectations, dispelling some of the crazy ones that the patients come in with, frankly, that they've read on Wikipedia or Google. And then the patient goes out of the room. We know that they remember probably 30% of what they've been told. And if if you have an optimistic disposition, you're probably going to remember all the wonderful things you think this new drug's going to do. If you're a pessimist, you're going to remember all the negative stuff. Two completely different expectations, same drug, you'll get two completely different outcomes. And so this combination of the pharmacology of the drug with how it's meeting the brain's expectations is actually what decides the outcome. So expectations matter. That's a setting up of belief states. As I said, the religious context is historically, a thing, but it's amazing how more we learn about how hardwired we are for belief and how it's such a core part of what it is to be human and how the brain works. And it does link to hope in a minute. You can see through these different experiments looking at different angles, we're all the time coming to the same neurocircuitry that's top-down controlling some of these things. That's very powerful. So what we did in this experiment is say, fine, expectations, beliefs, that's actually the fundamental thing that you're doing in a placebo manipulation. You set up somebody's expectations that this thing is going to produce this outcome. And you basically give them the experience of that analgesia so that they feel it, maybe with the drug or maybe by just turning the heat down. And then you actually do the manipulation with the sugar pill or the saline. And because they've been trained to do that, they've been what we call conditioned, they will mount that same response and get that pain relief, even to a sugar pill or saline. So you set up expectations, that's what you're doing. You set up the belief, and then you pull it away. And you can even do what I call placebo light. You can tell the patient, I'm giving you this sugar pill. It's got no pharmacological agent, but if you take it ritually, we know that studies have shown that if you take it and you really believe in the fact that this ritual is going to help you, you will drive. And you get, in irritable bowel syndrome, you get a significant benefit. It's not as good as the full Monty, full-blooded, full-fat placebo, but it works still pretty well because we're all capable of, again, tapping into these systems. So in this experiment, what we said, well, let's simulate what actually goes on in the physician-patient interaction, and that is we set up different expectations, but we give them the drug because that's the norm that happens. They come in, they get diagnosed, they get given the drug, but they'll walk out the room with different expectations. So why don't we control what those expectations are and see how powerful it is in terms of the drug? So what do? We take a bunch of students and we put them in the scanner and we're imaging their brain and we're asking them how do they feel to this thermal painful stimulus we're giving them. They give us our ratings. And they just know at some point they're going to get this drug through this intravenous infusion of one of the best analgesics we've ever had, called remifentanil, fast-acting mu opioid used in acute emergency medicine. It's brilliant. It works like a treat. We just don't tell them when we're starting and stopping it. So what we do is we burn away in the no opioid one baseline and we get their pain ratings. Then we start the infusion, but we don't tell them. And we burn away and we get their ratings. Then we do the big act and go into the room. The anaesthetist gets their old white coat on and goes in, right, we're going to start the infusion now. You're going to get that lovely analgesic. It's going to be great. And they think, oh, lovely. They've been on it for 15 minutes. They just didn't know it. So now we set up positive expectations. Do that. Then we go back into the room and say, right, over now. Sorry about that. We're going to turn the drug off. Back to baseline. Really sorry. And of course we don't. We leave it on. But they think they're not getting the infusion anymore. So we set up negative expectations. So we've got baseline. We've got hidden, no expectation. We've got open and positive expectation, and then we've got, again, a bit of a closed, which is negative expectation. They don't know what we're doing. They're just told to reliably tell me how painful the stimulus is. They don't know whether I've turned the heat up or down. We've given them exactly the same stimulus all the way through. The only thing we've changed, same drug infusion through, the only thing we've changed is expectation. And then we look at what happens to their ratings. So it starts off, it hurts. It's a good drug. So we start there, it's 60 out of 100, so that's sort of moderate to strong pain. That's the true pharmacodynamic effect of that dose of remifentanil. For any anaesthetist in the audience, thank God it works. You do get some pain relief. Now you add positive expectation on top of that, and you get double the analgesia. Just because of the positive expectation that it's been on, you get the same again. Now, what we hypothesized was we would reverse that gain when they thought the infusion stopped, but actually they went right back to baseline. They're still on the drug, it's still the same pain. That really freaked out my anaesthetists who were doing their doctorates with me on this study, because they're using this every day in their treatment, and they had no idea they had so much power over outcomes through the patient-physician social contract. It is huge. It is huge. And that's your chronic pain patient. This drug doesn't work, that drug doesn't work, this drug doesn't work, that drug doesn't. They just, it's a miracle anything has any effect when expectations have so mounted up to fail. And there's another story about how you need to use imaging for drug discovery, because if you were developing remifentanil and you looked at that, you said it didn't work if you didn't control your expectations in clinical trials. And of course then we're imaging and we're showing this isn't just that all the students worked out what I was trying to do. The brain goes down, it goes down more, and then we can work out mechanistically what's driving the extra positive expectation, what's driving them back to negative. And we can see that we've got our lovely little frontal cortex talking down to our brainstem that's driving them in terms of extra analgesia. And then this little area that was our sort of anxiety amplifier for other work we did way back in the day is where we got this anxiety amplifier driving them back to baseline pain ratings because they're anxious because they think the drug stopped when it hasn't. So this is a neural amplifier in the brain that makes pain worse. And other work we did shows if I just make somebody anxious, like going to the dentist, that region switches on. That's why the dentist hurts more than it should, because you're anxious. And anxiety it's literally a volume button inside your brain. And we worked out all the mechanisms that drives that. Okay, so this led then to this experiment, which was done by my very brave surgical colleagues, because we've been working with them on hip pain and looking at the bad cop and predictive of hip outcomes and knee outcomes in osteoarthritis. And then my dear friend and colleague who's running the orthopaedic hospital and department in Oxford, Andy Carr, brilliant shoulder surgeon, spent his whole career doing this shoulder surgery, which is this spur on the acromion joint, painful shoulder. He then, after lots of experience, said, you know what, I wonder whether my whole career doing the surgery is placebo. So he had the braveness and the courage to set up this trial. It took 10 years to execute. First of all, ethics were having none of it. Not as bad as the religious study, at least engaging with us. They didn't say flat no, but to get them to say we want to do a placebo surgery trial, we want to bring patients in, we want to bring them into the hospital, we want to pretend that we're doing a surgery, they didn't like that. So we had to walk them through why it's sort of ethical that we better find out. Because here's the thing that's different about surgery as opposed to drug development. Drug development, you can't get a drug on the market unless you've done your placebo-controlled, sham-controlled trial and check that you've got pharmacodynamic, efficacy over and above control. Surgery, you just go and do what you want. And if you want to tell them it works, you just sort of almost write a little protocol and you can have a go. And that's been great for other things that we've done with neurosurgeons to go in and turn things off. But if your surgery is an outcome that's subjective, like pain, I'm very happy that surgeons don't have to prove chopping out a tumour is a good idea. You don't need to do a sham control for me for that, or close the blood vessel when it's bleeding. But if you're doing a surgery where your primary outcome is pain, and you don't know pain is so subjective and malleable, you better check that the surgery mechanistically is doing something over and above that walloping placebo effect you can get, which is better than the drug, right? And they just haven't done it. So we did the study. It took 10 years, lots of different clinical trial units were in, then they didn't want to have the results, they didn't want to participate. This is one of the biggest private medical healthcare practices as well, certainly in the US. So you can imagine how popular he was and we were when the trial was done. We have a subset that have the imaging brain data, and the results are this. So what you've got is baseline, 6 months, 12 months. If it goes higher, it means it's more pain relief. Sorry, it's a bit back to front. And we had 3 groups. One group, nothing happened. They just carry on with your life. Well, even they improved after 12 months. But the red and the blue are the 2 groups that came into the hospital, had the full treatment of surgery, the anaesthetic, the overstay, all that. But only one group actually had the surgery. The other was a pretend sham surgery. No difference at 6 months, no difference at 12 months. It is a placebo effect. It's now been replicated, and we've written statements and reviews, and the team that led on this has written statements on how one needs to go through all the different surgeries for the treatment of pain and make sure that you're not just manipulating people's expectations and beliefs. Now, that's fine. If this is what you need to do in order to get that effect, go for it. But let's just be honest to the patient and the hospital about the cost, the risk of coming into the hospital with all the associated risks. You know, there's some interesting ethical questions there. If this is what you need to do to get that pain relief, fine, but let's be really clear, it's an expectation belief change. It's not a mechanistic thing. And so that's plodding through different surgical treatments for pain to see whether they are mechanistically doing something or whether it's a placebo effect. But I share that with you as just a sort of nice translational example of where we're going. So these beliefs, these things, we're very much, for those of you who are sort of into belief, and I've mentioned a lot about the sort of importance of how we're wired up to believe, it's fundamentally more and more how we actually think the human brain works. So the very, very latest way we think about how the human brain works is it's not just in this side of it, a receipt model where you have this organ, it receives things through your sensory organs, like your eyes and your ears and your nose and your taste and your mouth, your skin. So, you know, the sort of original textbook model, it's sitting there waiting for these inputs to come in. It processes them. You realise, oh, I heard something, I smelt something, I saw something. What does that mean? Oh, I'm going to react back out. That's how we sort of teach it, but it's not how the brain works. You don't have to think for a minute. That's a really inefficient way for the brain to work, because all day long you've just been bombarded with all these inputs and then working out what they mean. So what you do is you slightly short-circuit it. Because of that evolution and that development of your brain, we now think of the brain as a Bayesian brain. This idea based on Reverend Bayes, who had this idea of probabilities, that what happens here is contingent on what's happened before, and then it happens again, and you can load the dice to sort of show how the next event is going to be a reflection of this event, and you build up a probability or prior expectation or belief about what the next state's going to be like based on this state. And we now know that the brain probably works like that. So often the analogy I'll use, normally with a glass of water or something, is, you know, well, I'll touch this thing. It looks like it's metal, so I'm going to touch that. I can see it's metal. I'm 58 years old. I've touched a lot of metal objects, so I've already decided in my brain what that's likely to feel like. I've got already a network in my brain that's prior as to what that sensory experience is going to feel like. I now touch it, and I have the sensory transduction and transmission thing going, but it is meeting a brain that's already got a prior expectation or belief about what that's going to feel like. And it's the combination of those two things that actually gives me my perception. Suddenly that changes everything about how we look at people in the world. Because if the prior is really strong, because you've had lots of rehearsal, probability experiences that these things feel like this, it will become the dominant influence on what the perception is, irrespective of what's coming up the spinal cord. So if you know that every time you get up, your hip's going to hurt, and that's gone on for 10, 15 years, but you want to go make a cup of tea, well, maybe that one time you got up actually didn't twinge the nerve or whatever, but your priors are telling you it hurts every time you do that particular thing. You've built up that expectation, that belief. So you will have that experience because your priors and your network will set them off. And the bottom up becomes almost an irrelevance. Panic disorders. Doesn't matter what data you give people about their heart rates not changing. They think they're having a full-blown heart attack. It's a prior. It's just a disposition of the brain. It's hardwired in there. So suddenly it starts to explain, you know, hysteria, all these other conditions that have been binned as, you know, in the mind and people weren't real. Now we've got a working model of the brain that's very, very different and can get us to think quite differently about, again, not just conditions, but just how do we fundamentally want to approach things like beliefs. And it gets at the core. So this is just, if I took you again in the scanner and I put you in my scanner and I had you just do absolutely nothing, eyes closed, nothing to hear, try and not think about what you're going to have for dinner tonight or what your worries are, really try and just go into, you know, chill. You'd think that we'd just be measuring a chugging little brain just chomping away on a bit of oxygen and glucose. But this is what your brain actually looks like in this condition. Oops. Mm-hmm. What you can see going on there, lots of different brain activity. You can see there's colour-coded ones. Some are in green, some are in blue. Each one's colour-coded for a network that's active. And when we look at those networks, what we see is it's your memory network. your vision network, your auditory network, your touch network, your motor network, all the networks of the brain that allow you to do movement, hearing, seeing, tasting, decision-making, thinking, cognition, all that sort of stuff. Even when you're not doing any of those things, they are preferentially having a conversation with each other. They're on. They're on even at rest. They're ready to go. So it's a way to sort of just illustrate, well, one, your brain's really busy even at rest. If you fire together, you wire together, that's the thing. So even if you're not using it, it's preferentially coordinated and having a conversation ready to go, just in case you need to feel something or hear something or see something. You short-circuit that because it's a more efficient way for the brain to work. I hope that sort of makes sense. So this opens up all sorts of different ways for us to think about, again, the interaction of that and pains and beliefs. You know, we get to the sort of final point, which is really sort of, as we think further about, I get asked a lot, can robots feel pain? Is there consciousness in the brain? I'm skipping over, we've done lots of work on consciousness. Those are hard questions. And of course you can build a robot to produce a response that will look no different. I can train it to look as if it's experienced pain in the classic way one would. But of course I don't know, just like I never really know what your pain is, and you'll never know what my pain is. I never really know what's going on there. And I do believe there is something that is different, obviously exquisitely different, about a sentient human who is not just a brain and an organ, which is obviously what we focused on today with the imaging of it. But of course the brain exists in confluence with your body, and a sentient body. And to be human is different. And to value something that the humans produced it, even if it might not be as good as what will be the future AI and art, we'd have a different way that we value things, which come back to sort of some very deep things about how the human brain is just wired up and how we evolve that brain. It's not going to be short-circuited very soon, I don't think, even though there's a lot of excitement around AI. And of course, you know, again, its biggest challenge will be how does it make that transition from just being a computational thing into that realm of these other things that are more important about what it means to have respect and empathy and dignity and theory of mind and all those sorts of things. That's very natural for a human to be able to go into somebody else's brain and think about how they might be. So we can manipulate things, we can do the best we can, but, you know, I still have a lot of hope for humans, although we're doing a lot on AI, obviously, back in Oxford, and ethics of AI as well. A lot of philosophers thinking about how do we live alongside these things. I just going to finish on this one. I said, mentioned I was, you know, I'm practicing Roman Catholic. I was very blessed. You mentioned the guest edit I did for the Today programme between Christmas and New Year, and they wanted me to sort of, I talked about pain and what universities do in its broader context, and we went up to Islay actually and looked at how we make malt whiskey, which was fantastic fun because they wanted a bit of fun with it as well. But they wanted me to also sort of illustrate how, you know, that I am a person of faith. And I joked and said, oh, I'll get the Pope to do the thought for the day. And they're like, would you? And so anyway, I managed to do it. So it was his last ever BBC thing, and he did it. And my theme for the guest edit was hope and kindness. It's something I've been banging on about in the university and trying to get students to think about the importance of kindness and the strength and etc. And I recently did a speech on this topic. So he did a beautiful, beautiful thing on hope and kindness. And of course, it's the Jubilee year for the Catholic Church this year, which is all about hope. And us to be pilgrims of hope and to bring that to people. So in some recent work, I've been exploring a little bit, just for speeches, not for research I'm doing, just about the sort of neuroscience behind hope and kindness. And what's been, again, in a more social neuroscience, some of the developments there that, again, get at these really interesting intersections with what it is to be human, what separates us from artificial intelligence as opposed to natural intelligence, what it means to be in a society. And it's really quite exciting, this sort of intersection between what we're learning about the neuroanatomy and neuroscience of belief, looking at it from the different ways I've illustrated, about hope having this very core sense of being able to have a goal, being able to have a path and a process to achieve that goal, and having the agency to affect delivery of that goal. It's really important for humans and society to have hope. Because it's actually what evolves us and what drives us forward in society. Look at how many times we should have blown up this planet and yet we haven't. And we all know how desperate it can be for colleagues or friends or students if they get into a state of hopelessness where they feel they've got no agency and they've got no path to a better goal. These are fundamental things that are very, very deep in some of now our understanding of how the brain works, that we can start to tackle that and start to think about how we can bring more benefit. And kindness is another one. And I stress this a lot with students and faculty, is the importance to show kindness. It's a great strength to be able to be a kind person. It's not a weak thing, it's a very powerful thing. It's a very important thing, and we need a bit more kindness around. But of course, to be kind again, it taps into these deep elements of what it is to understand theory of mind, to be able to go into somebody else's mindset and see how are they probably feeling in this situation, what empathy do I have, and then to display some compassion. And it releases oxytocin, it activates reward centres, it brings, we know that generosity through kindness can bring a great sense of satisfaction and reward. And there's a really healthy neuroscience now going on in these two elements that's sort of helping us understand a bit more about what the neuroscience basis is of some of these things that, again, start to become intertwined, I think, with the sort of concept of belief. So with that, I will now finish because I've gone on and on. But if you want to sort of follow up on some of that, as mentioned, I've done quite a bit of public understanding stuff. But the main slide is this one, which all the wonderful people I've had the very, very good fortune of training and working with and learning from, most importantly, and becoming, just colleagues and friends for life as they develop and grow and have families and develop their careers. We are very blessed to be academics, those of us in the audience who are academics, and it's a wonderful, wonderful career. I know it's not easy. It's not an easy gig. It's a hard job, and we demand a lot of you in terms of the research and the teaching and communicating. But it's an extraordinary privilege when you look back in your arc of life, to just see the different things that one's been able to discover and the different people that you've been able to influence just in a small way. So I want to finish by thanking all my wonderful former and just finishing off current team members as they finish through. And of course, we can't do any of this unless people pay us to do it. So thank you to all my funding agencies. But mostly, thank you for the honour of coming again to this wonderful university. I wish you all the very, very best going forward, all the wonderful, important work that you are doing. It'd be lovely to talk further about how we might be able to collaborate. And I hope that I've inspired some of you to think about reaching out to your neuroscience colleagues and seeing whether there's some questions that they might want to pursue informed by you and your knowledge. Thank you.